Oxidative phosphorylation, in plain English. This presentation covers one of the major processes carried out by microorganisms and other life forms to create adenosine triphosphate, or ATP, which is the usable form of energy. All living things make and use energy sources, including this little microbe, prokaryotic organism. These organisms use energy in the form of the aforementioned ATP, which stores energy in its electron bonds at the atomic level. In this presentation, we will focus on aerobic and anaerobic oxidative phosphorylation, which occurs in the cell membrane of prokaryotic organisms. In all energy transfers, the net charge must be conserved. Therefore, in the process of oxidative phosphorylation, we have an electron donor, NADH2, and we also have electron acceptors, like NAD+. And also, importantly, the terminal electron acceptor, oxygen. So the oxidative portion of oxidative phosphorylation depends on the electron transport chain, which of course begins with electrons. Electrons are passed from the electron donor to a protein complex spanning the phospholipid bilayer called dehydrogenase. The electron donor is thus oxidized to its electron acceptor form, NAD+, while dehydrogenase is reduced. The dehydrogenase complex uses these electrons to pump protons across the cell membrane to the outside of the cell, or periplasm. This proton transfer begins to establish the proton motive force, or increasing concentration of protons outside of the cell. We will return to this concept. Now, the electrons will move on to the next station of the electron transport chain, which is the molecule quinone. Quinone is reduced to quinol by accepting these electrons, which can now pass them on to the next part of the electron transport chain. Most microbes have the cytochrome Bc complex, which accepts the electrons from quinol. Cytochrome Bc then pumps protons across the cell membrane, like the previous protein complex, dehydrogenase. Once again, this increases the concentration of protons outside of the cell, further building the proton motive force. The electrons are then passed on to their next destination, cytochrome C, which is subsequently converted to its reduced form. Cytochrome C doesn't waste any time passing the electrons on to their terminal protein complex, which is termed cytochrome oxidase. As before, cytochrome oxidase pumps protons across the cell membrane, which finalizes this highly concentrated collection of protons outside of the cell membrane. This concept of proton motive force will be very important when discussing the actual phosphorylation portion of oxidative phosphorylation. Cytochrome oxidase, however, is the end of the electron transport chain, and it must pass its electrons somewhere, so they are transferred to the terminal electron acceptor, which is oxygen. The electrons from the electron transport chain are passed from oxidase to oxygen. And now protons still within the cell are attracted to these outer shell electrons of the oxygen atom. And these three atoms come together to form a water molecule, ending the electron transport chain. Now for the actual phosphorylation of this dynamic process. So how is ADP and a lone phosphate group transformed into ATP, which is a usable source of energy? It all begins with the previously established high concentration of protons outside of the cell and this membrane protein complex, ATPase. ATPase allows the protons outside of the cell to come streaming through its inner chamber. This equalizes the proton concentration gradient, and the energy change of these protons going from a high to low concentration converts into electrical energy in the form of those high energy electron bonds, which fuse ADP and the lone phosphate group to form ATP. Before, the concept of aerobic oxidative phosphorylation was discussed, as it used oxygen as its terminal electron acceptor, and oxidase was the terminal protein complex. Now we'll discuss anaerobic oxidative phosphorylation. In anaerobic environments, we encounter protein complexes such as nitrate reductase, which is an alternate terminal complex. After electrons travel through this complex, They are passed to terminal electron acceptors such as nitrates or sulfates, which are then, of course, reduced to nitrites and sulfites. This process, of course, produces ATP, just like aerobic oxidative phosphorylation ultimately. However, it does not do so with the use of oxygen. In conclusion, these are some of the processes that govern how microbes can produce energy in the form of ATP. Now the microbes can use this energy to perform daily tasks like chemotaxis, as observed here, allowing these small microbes to propel themselves with their flagella through the harsh environment of the prokaryotic world. The end.